Hey, Phil, I have to teach transpiration online. Any suggestions?
What we're doing is we're taking an existing transpiration lab that we use here at the University of Oklahoma and modifying it for online teaching. The way that this lab normally works is we give students a protocol online that explains sort of the general setup of the lab, and then they would show up and do that. But right now, that's not really possible because of the situation. So what we've done is we have students still looking at the protocol beforehand, but we've made a video that we posted on my website that gives the general outline of the experiment that we'll be doing. It shows them the plants that they would have been working with and how the experiment's set up, and then walks them through sort of what they would have been doing, explains the kind of manipulations we would have had there, and then shows them some data or tells them about some data that they can work with. And so that's what we're doing to talk about transpiration in my class.
Okay. So typically, what does this lab look like? I mean, if they were in lab, what would they be doing?
So normally what would have happened in lab is that they would come in, we would give them in their groups a certain number of plants. These would either be corn seedlings or sunflower seedlings, so a C4 and a C3 plant respectively. And they would take the seedlings and place the pot inside of a plastic bag and then tighten it with close it up with a twist tie around the stem and take an initial weight of that plant. What they would then do if they're in one of the treatment groups or even in the control group is have their plants and weigh them at different regular time intervals. So for example, the control group just takes their plants, sets them on the lab bench. The treatment groups might take their plants and put them in front of a fan or put them in darkness or put them under some different colored light. But then we take measurements every 10 minutes or every 20 minutes, sort of depends on what your lab time period is that you're working with. And over that period of time, the plants are transpiring water out. So as we weigh the plant, that plant in the pot at time point, what we're getting is a measurement of how much water has been lost out of that plant.
Okay, great. And so you have these data sets put together for the students to work with online?
I have a data set that we've shared with our students. I can call that up on my screen. Let me share my screen. I can show you what those look like.
Yeah, please show me.
So here's the basic data set. These are data that my TAs pulled together from previous semesters. And what we have are some data from the light manipulations here. So we have the weight of 3 different control plants at 20-minute intervals. So we run this for 100 minutes, and you get nice responses from the plants in that time. I should also add that before the lab starts, all of the plants have been well watered, so they aren't water stressed to begin with. But you can see here the volume of water that plant has transpired, just basically the weight change there. We calculate the percent weight change in the plant. And you want that percent weight change in there because you've got to standardize your data somehow. You're going to have different size corn plants and sunflower plants. So by looking just that overall percent weight change, everything's evened out. So you can see in the data set we're sharing with the students that they've got their data from control plants, or sorry, C3 plants grown in blue light, C4 plants grown in blue light, and so forth. And just to help things along a little bit, we summarize some of the data for the students. One of the challenges we've got now is we can't be right there with them, showing them how to do the calculations. So we're doing some of this, just kind of lead them along and help them. So they have some summarized data here. And then over on the side here, we've summarized the percent weight changes as well. Down below, we have, again, control data. And then these are plants that we added salt water to, as well as plants that we put in the wind. The wind obviously is going to drive more evaporation, you would think. And so will the plants, just allow the water to evaporate? Will they close their stomata? That's an interesting part of this experiment. Oh, wrong screen. And then the salt water, that's a fun one because transpiration is driven by gradients in water potential. So by putting salt water into the soil, what you're doing is you're changing that balance of the water potential gradient. And so it's harder for the plants to pull. And so we wanted to see if that would influence the transpirational rate because it should pull it down. So these are the data that we've shared with our students. Let me pull up now the TA data. So here, I've got a combination set of data. This has everything. It's basically the same setup for the TAs who help me teach the lab, but I've gone ahead and graphed out the data. So here you can see our control sunflower plant, our control corn plants. And this is what happens when we put the plants in red light. So you can see how the transpiration really picked up early on for both of them, but came to about the same level after 100 minutes. Blue light was cool to look at. It still got some transpiration. It's still stimulating the plants to do photosynthesis, but at a lower level than the full white light or even the red light. Green light was really cool. This is always a fun one because you can see how the transpiration just bottoms out. There's really nothing happening. And this is awesome when you compare it to plants in the dark because you get the same thing. And that's a really good moment for the students to see that, you know, you're growing plants in green light, or if they get a lot of green light to plant, they're in the dark. You can't see anything. The wind, pretty much what we expected. One thing that if you do run this out longer is you'll see the C4 plant, it'll shut down. They have a lot more stomatal control, and so you'll see it really regulate its transpiration more than the C3 plant. And the really cool one is when you water them with salt. As you'd expect, because you put the salt water on, it's going to have an effect on its water potential. You can see that the sunflower plant here in the control, it just can't pull out that extra water. Same thing here with the corn. So the experiment works great. It's very reliable. And here we made, sorry, here's another chart. And you can see that the volume transpired by the control, the percent weight change, and then looking at all these different treatments. So there's a lot of good data here. It's a very data-rich exercise, which is one of the reasons we use this in my class, is it gives the students a lot of data to work with. The experimental results are very clear, straightforward. It always works. Like I say, the plants know what to do, and so that helps. But it's a wonderful experiment, and although I wish the students actually had the chance to work with the plants, and it is kind of nice because the students like to keep the plants after the experiment. So you'll see all these little intro biology students walking across campus with sunflower plants and corn plants and stuff. They didn't get to do that, but we still give them the same experience. They're getting the data, they're getting the analysis, they're still writing the assignment, which is to produce an abstract. No matter how we had done this, we would still say that, okay, you have your data, you've done your experiment. What they do is write up an abstract to summarize the experiment, and then they can also, well, they have to include a figure with it. And the data are very nice and clean, and it works out really well. So we still get the same type of experience working with data that we would have had before. It's just they don't get to actually do the hands-on part with the plants. But we can still make it work. It's still a good botanical experience, I think.
Yeah, that's great. How long are you giving them in class to do that?
Well, so normally this would be a one lab period. So if our labs are 3 hours, and they can get all of this done typically, you know, doing the experiment, writing up the abstract, and submitting that in a 3-hour period. We're dealing with a whole bunch of different time frames and time experiences now. So we're giving them 2 weeks to work on this. So this week we're introducing it to everybody, showing them what's happening. So the TAs are still meeting with our labs for an hour. I think the TAs have said it's actually taken only about a half hour, but they explain the experiment, give them the data, tell them what's expected. And so the students, we're hoping that they'll go work with it a little bit over this next week. They'll meet with them again next week. And if they've had any problems or have questions, they can ask their TA about it then. And they've got another week to wrap it up. So we're giving them plenty of time to work on it. We told the students that they can work together. They can have Zoom meetings or whatever, but we want them to write it up individually. We know all the figures are going to look exactly the same, and we don't care. We know that's what's going to happen. We're kind of hoping the figures will look the same, and that's why I gave my TAs the data set with the figures already, so they have a nice key that they can use to start looking at all of these assignments that'll be coming in.
Okay, great. That was very helpful. Thank you very much.
Thank you.